In this video, we will discuss about this FASL gene that is overexpressed in the TME or in the tumor microenvironment. So this research is also in line with this FASL expressions and its impact on the T cells survivals. So first they check the data that is available in the literatures and compare it with the normal tissues data, and they are able to see that in most of the cancer cases the expression of FASL genes is overexpressed. In terms of the quantifications, they can see that 19 out of 26 that they have tested has this overexpression of this FASL genes, and around 5 out of 26 are non-significant, and 2 out of 26 has underexpressed. So once they have some preliminary analysis that FASL genes are overexpressed in the cancerous tissues compared to its normal tissue, they check the RNA-seq analysis data of these different types of cancers that is publicly available, and did the genes and enrichment analysis to find out what are the pathways that are highly upregulated. And they can see that natural killer cells mediate cytotoxicity, which is expected, and this antigen processing and presentations, the T cell receptor pathways, this primary immunodeficiencies, and they also see apoptosis, and this apoptosis could be the apoptosis of the T cells also. And thereafter they check, once they have this some more results from the gene cell enrichment analysis that supports their hypothesis that FASLs might trigger apoptosis. And now they wanted to check the top genes that are related to the FASL genes and its impact, or the pathways that are related to those top 200 upregulated genes that is related to the FASL gene. And they can see that those upregulated genes are the markers for the immune response, such as IFNG and PRF1 and the IL-10RA. So from that they come to a point that FASL's upregulations might also be related to some kind of immune response, as the high amount of FASL genes is correlated with the high amount of the markers that might trigger immune response. So once they have some of the they have all these results, they wanted to check in the patients and in the healthy donor the expressions levels of the FASL receptors on the T cells. So they took the samples from the healthy donor and also from the patients who are affected with the cancers, and first they check the CD8 different subset of CD8 plus T cells. It is T central memory, T effector memories, and TEM arrays. I'm not able to recall its complete full form. And they can see that all three subset of T cells, CD8 plus T cells, are found in the patients. And then within this subset they check the FAS expressions, and they can see that FAS expressions in all the activated T cells, that is T central memory, T effector memory, TEM arrays, are higher than the T neutral cells or unactivated cells. So this might indicate that in the activated cells somehow these FAS receptors get expressed on the cell surface. And they also did one more analysis to check, and that they have find out is about the percentage of the T neutral cells in the healthy donor and compare it with the donor who are affected with the cancers. And they can see that donors who are affected with the cancers has low percentage of the T neutral cells compared to the donor who is healthy. And the reasons that the researchers have cited that this could be because the patients have undergone multiple immunotherapies or multiple kind of therapies that might cause the depletion of the T cells or the immune cells within the patients. And this also in line with the patient's history that they have undergone some kind of therapies. So from these results they finally they come to a point that FASLs are overexpressed in the tumor microenvironment, and that might interact with these FASL receptors. So that interactions results are not shown here, but this is what the hypothesis that come to the researcher's mind, because FASL genes are also overexpressed and FAS receptors are also overexpressed The difference is that FASL receptors are overexpressed on the T cells and FASL genes are overexpressed in the tumor microenvironment, and there is a high possibility that both of them can interact, and it might cause some of the results that might it might cause some of the signaling pathways activations that might result in the apoptosis of the T cells that we have discussed in our previous papers that when we are discussing our previous papers. So far they have just presented here that FASL are overexpressed in the tumor microenvironment and FASL receptors are overexpressed on the T cells. That's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.